Good afternoon. My name is Jaime Guidry Auvil, and I am a Scientific Program Manager in the Office of Cancer Genomics at the National Cancer Institute. Today I'll be talking to you about targeting better treatments for childhood cancers and our Pediatric Childhood Cancer Genomics Initiative, the TARGET program. I have no disclosure information. So this here is a chart that you can see is U.S. childhood mortality trends for lymphoma and leukemia versus all of the cancer sites combined. It was from JCO, published in 2010, and one of the co-directors of the Target Initiative had published this. It's a colleague of ours in CTEP, and this is indicating that since 1975, for about 30 years or so, trends in childhood cancer mortality rates were diminishing greatly. And this was more pronounced for leukemia and lymphomas. However, this is true of all childhood cancers. What's interesting to point out is that since the early 2000s, this trend has plateaued. And we see that treatments that were working and were reducing mortality rates are no longer trending in that direction. And things are fairly stagnant. So it's become obvious that there is a need for new treatments in childhood cancer across the board. So this same group that had published this data had gotten together around that same time a few years previous and noted that in order to change the trend of childhood cancer mortality, that we needed some new, more effective treatments. This is important for many reasons. Childhood cancers are generally treated with adult regimens that are dosed down or scaled down in the dosage or in the timing to account for a child's body and to be able to handle that. This is not always the best option. These treatments continue to be very toxic, and so even though children are surviving, they are maintaining long-term side effects and high morbidity. That includes increasing the risk for secondary cancers, as well as infertility, continued pain, and learning disabilities and development. And these things need to be addressed as much as the need to reach those who are not surviving childhood cancers. So the hypothesis that led to the development of the Target Initiative, which stands for Therapeutically Applicable Research to Generate Effective Treatments, was that the genes or pathways that are consistently altered in specific childhood cancers are central to the discovery of new therapeutic targets and new treatments. And secondly, that these insights will be one of the key drivers of progress in identifying more effective treatments for children with cancer. So the strategy for discovering effective new treatments in children with cancer is meant to begin with our program, which is a genomics discovery program, to find mutations that are drivers that might also be targetable with different drugs that are either existing or could be developed. Now, this program is meant to go quickly through the steps of preclinical evaluation, pediatric phase 1 trials, and then to positive definitive clinical trials that will translate to clinical care. So the target strategy was to partner with the Children's Oncology Group, which is a cooperative group that has a good system in place to obtain clinical samples from their biology and treatment trials. They are possessing rich clinical annotation so that outcome measures can be determined. There is an emphasis on cases with high risk of treatment failure, as we saw earlier. Standard risk kids, or kids with cancer with standard risks, are actually faring fairly well on current treatments. However, kids in the higher risk treatment failures are succumbing to disease. We also wanted to aim for 100 to 200 cases per disease, and that would have to include tumor and matched normal samples. We also decided that it would be good to work in a collaborative network of team science to more effectively get things done. So disease specialists are working from within the COG, also with colleagues at NCI, to be able to accomplish these goals. And the project teams will be analyzing comprehensive characterization molecular results along with counterparts at the National Cancer Institute. And of course, NCI can play the role of running sequencing contracts and funding those to help further the science and the type of sequencing that was hypothesized could be done would be not only targeted Sanger sequencing, but next generation sequencing to include whole genome, whole exome, and mRNA-seq. And most importantly, is that the characterization and sequencing data that such a project would generate is made available to the research community with the clinical annotation so that further studies can be done, thereby leading to more effective treatments in a more timely fashion. So a pilot was anticipated. This is a map of what the target pilot started out as. I will be telling you a little bit more about that. It began with acute lymphoblastic leukemia and then moved into a pilot in the neuroblastoma disease. So as I said, the ALL pilot was the first one to be tested with this new sequencing and characterization initiative. The goal of that study was to characterize 189 patients. They ended up being from COG trial P9906. This included high-risk precursor B-cell ALL patients who relapsed within 36 months and also excluded most recurrent translocations with the exception of E2A, PBX1, and MLLR. There was to be array-based characterization on these samples to include gene expression and copy number, and next generation sequencing that included 20 whole genomes done with Illumina and some with complete genomics, as well as 10 transcriptomes, in addition to Sanger sequencing that included targeted resequencing of 125 genes and also some kinome sequencing. This study is complete and all data is available at this time. Just to briefly show you what I was iterating before is that you can see here on the left-hand side that newly diagnosed ALL has a good prognosis overall, and most children are recovering from the disease. However, those kids that do happen to relapse, you can see have a very, very poor prognosis and a low chance of survival with only 24% as compared to more than 90%. And it's this group of cases that we have chosen to focus on in the target initiative. So some major findings from the ALL pilot were that BCR-ABL1-like ALL, that it is a phenotype that exists with Ikaros deletions and mutations. This is all published. It does have a gene expression profile similar to Philadelphia chromosome positive or BCR-ABL1 positive ALL. It comprises 10 to 15% of childhood BLL. The incidence is higher in young adults. High frequency of Ikaros deletion or mutation, as I mentioned, and an extraordinarily poor outcome. We have also found that JAK mutations are prevalent with high-risk ALL, and that 50% of those also have CRLF2 alterations. We also have found that recurrent mutations in 4 key pathways seem to be present, and the pathways include B-cell development or differentiation, P53RB tumor suppressor pathway, RAS signaling, and also Janus kinases. The BCR-ABL1-like cases with somatic mutations in B-cell development are extraordinarily prevalent at about 96%. However, JAK mutations and P53RB mutations are also prevalent in more than half of the cases. We have also found that there are kinase activating lesions, but no point mutations in BCR-ABL1-like B-cell ALL. Other tyrosine kinase gene point mutations are rare. High frequency of gene fusions targeting ABL1 and ABL2, CSF1, JAK2, PDGFRB are also partnering. And ABL1 PDGFRB fusions highly respond to ABL tyrosine kinases in vitro in xenografts and anecdotally in humans. These are being tested currently. So some downstream publications are listed here. There have been quite a few publications now to come directly from the target project team, more about the CRLF mutations in Down syndrome patients specifically, as well as expression profiling. And the research community, because this data is made available to them, have also been publishing. Due to the positive results of the ALL project, another disease project was determined that to be added as we had some funding. So neuroblastoma was the second disease chosen, and you can see here in this graph for the same reasons that both low risk and intermediate risk cancers in neuroblastoma tend to fare fairly well. There's a high event-free survival, whereas the high-risk cases, for whatever reason, really are not treatable on current protocols. So the high risk includes stage 4 patients, generally between 18 months and 5 years. The Neuroblastoma Project sought to characterize about 180 of these cases with full characterization, but there are an additional 420 plus cases with partial characterization, and a subset of those are low risk as comparators. They too were using both array-based and sequence-based characterization to look for driver mutations. And they did gene expression and copy number as well. They also had some targeted resequencing of 116 genes, as well as the whole genome and transcriptome sequencing seen in ALL. But in addition, they were able to sequence 221 whole exomes to add for information from the brood. This study too is complete, and all data is available. It was published earlier this year in the genetic landscape of high-risk neuroblastoma. The findings really were that there's quite a low median exonic mutation frequency and very few recurrent gene mutations seen in the cases that were analyzed. And here to the right, you can see surface plots that depict the fact that there are very few rearrangements found in neuroblastoma. Genes with significant mutation frequency include ALK, PTPN11, ATRX, MYC-N, and NRAS, and that rare pathogenic germlines are enriched, including again in ALK, CHEK2, PINK1, and BARD1. So some translation of the target ALL findings into clinical trials for acute lymphoblastic leukemia include a COG phase 1 trial on Janus kinase inhibitor, ruxolitinib, in patients with CRLF2 and JAK mutations. And there are also a phase 3 trial that's just beginning, studying the efficacy of combination therapy in treating young patients with high-risk BALL. And also in neuroblastoma, a COG phase 1 and phase 2 trial of ALK inhibitor crizotinib. The first publication on that has already come out in May by Yael Masi and colleagues in Lancet Oncology, and you can find out more information about these at clinicaltrials.gov. So based on these pilot findings, the American Reinvestment and Recovery Act, or ARRA, provided some funding to actually expand the target initiative into what it is currently today. So as you can see here, this is a schematic of how the current target program works, and this is based off of, again, the successes that we had in the pilots of ALL and MBL. So we are still using disease expertise in team science, both within the project teams and with NCI. There is transcriptomic filing, both chip-based and/or sequencing of the transcriptome, mRNA and miRNA. There is genomic characterization, also chip-based characterization of the genome and epigenome, and gene sequencing, whole genome and or exome sequencing. And as you saw, there was targeting sequencing in the pilot that is available now. All of this is being performed on richly annotated, high-quality specimens. Our current target initiative consists of 5 disease project teams. We have a disease project in ALL, a disease project in AML, neuroblastoma, osteosarcoma, and kidney tumors. It's important to note that we are still partnering heavily with the COG and both the Biopathology Center out of Nationwide Children's Hospital in Columbus works closely with us, and the COG Group Operations and Cog Statistics and Data Centers out in California provide much of the outcome measures on the samples provided by the Pathology Center. And now you can see that the target collaborators map has grown quite a bit. Our project teams are spread out throughout the country and in fact somewhat internationally at this point, but most of our cases do come from COG here within the United States. So to talk a little bit about the current target initiative and the data that will be coming out later this year in 2013 and early 2014. This is the Neuroblastoma project that I was already describing, but the ARA funds did allow it to expand a bit. So using mostly the same cohort of roughly 600 some patients, we were able to fully comprehensively characterize about 210 patients with neuroblastoma. These again were stage 4 patients enrolled in the biology trials from COG. About 10% of those cases we were able to get as 4S cases, which start out appearing to be high stage, but then spontaneously regress. And then we also have about 5% of these cases that were relapsed cases where we have trios, which is not only a tumor normal sample, but also the relapse sample, where we can get a more rich outcome. The project was expanded to include not only the gene expression and the copy number that was already existing from the pilot, but also to gain methylation profiling on a subset of the 210 cases, as well as a very rich amount of next generation sequencing that includes whole genome and mRNA-seq on 3/4 of the cases involved, and up to 30% of those cases will have whole genome, whole exome, and mRNA-seq, so it's going to be quite an exciting data set. We have another ALL project. It is the same project team as the pilot, however, it is a brand new cohort of patients. So again, focusing on the high-risk BLL patients, the group actually selected cases that had come from both standard risk and high-risk protocols, but had relapsed within 48 months of diagnosis. About 40 to 45 of those cases are trios, again, that have a relapse sample in addition to tumor and normal. This group will have gene expression, copy number, and methylation through the HELP assay on a subset of cases for array-based characterization. All cases will have whole genome sequencing through complete genomics. A subset of these cases, and some with relapse, will also have both mRNA-seq and miRNA-seq, and about 10% of those will have whole genome profiling or whole exome, profiling as well. Acute Myeloid Leukemia Project is similar to those two mentioned. They are comprehensively characterizing about 200 high-risk AML patients. Half of them are trios with relapse samples. They too will have gene expression, copy number, and methylation array-based characterization. All cases will have whole genome sequencing through CGCI. All cases for AML will have mRNA-seq and miRNA-seq. And again, 10% will also have whole exome, and those are primarily trios. The AML group was also able to find 30 cases of AML that failed induction, and we are going to be characterizing them with whole genome sequencing, as well as RNA-seq and microRNA-seq of the diagnostic tumor, of the failed remission sample, and a cultured fibroblast as a normal control. A kidneys tumor project was able to be begun. The ARA funding allowed for high-risk Wilms tumors to be studied. This is a tumor that is generally thought of as a very treatable tumor. However, there are two cases in which the patients do not fare as well, and that is if they are anaplastic or favorable histology tumors that end up relapsing, and we don't know why. So 33% of the cases, of the 120 cases that are going to be studied in Wilms tumor, are anaplastic, and about 2/3 of those are the favorable histology that have relapsed, and 5% of those that have relapsed, we will have the relapse sample as a comparator as well. Again, array-based characterization does also consist of gene expression, copy number, and methylation, and next-generation sequencing. About 65% of these cases will have whole genome sequencing. All cases will have mRNA-seq and miRNA-seq, and about 35% will have whole exomes. With some additional funding, this group was also able to characterize clear cell sarcoma of the kidney and rhabdoid tumors. There's about a dozen cases of CCSK, and they will have both array-based and next-generation sequencing, primarily whole genome. And the rhabdoid tumors, there's about 40 will be characterized comprehensively. This is will be a very interesting data set, particularly when that's made public, because it is a full sequencing data set of whole genome, mRNA-seq, miRNA-seq, methyl-seq, and ChIP-seq. And we also will be including some cell lines as well as human embryonic stem cell lines. And finally, our last of the 5 major project teams is an Osteosarcoma Project. This group will be characterizing about 95 high-risk patients. These are mostly COG cases. However, we do have some external collaborators to add samples. The osteosarcoma as a disease is extraordinarily difficult to get the quantity of DNA and RNA that's at a high quality for these cases, simply because of the nature of the disease and how the bone has to be processed when extracted. But so this will be also a very rich data set that really has not been done before. There is array-based characterization of gene expression, copy number, and methylation like the other diseases. This is the one group that is actually doing a chip-based characterization for microRNA. And then there will be next-generation sequencing of whole genome, mRNA-seq, and all cases in osteo will have whole exome. So this is a summary of basically what I have told you all already, but you can see how much information is out there versus what is coming. So you can see that most of the groups have completed the next generation sequencing that they hope to have completed on their samples, and manuscripts are in process now. There is quite a large amount of whole genome sequencing, which is unique to the target program, and there is also a fair amount of exome sequencing as a comparison. And the determinant for us of which was a possibility was really based on the amount of DNA that was able to be used. But each sample obtained as much sequencing as was possible so that we could get clear results that hopefully can be turned into effective treatments for these children. And here is just a little snippet of what's to come. These are Circos plots from whole genome sequencing, and you can see in ALL, AML, and MPO, we have some good primary tumor versus relapse comparators. And you can see, again, as was pointed out previously for neuroblastoma in the pilot, that there are relatively few genomic rearrangements in most of these childhood diseases, with the exception of osteosarcoma. Osteosarcoma is a highly rearranged genome, and we like to call that the angry genome. But there is certainly quite a lot to study there, and this data, again, will be available later this year or early in the spring of 2014. So this is a list of the pilot project publications, all of which have been discussed previously, but I wanted to remind everyone that these are data sets that are currently available to the public, and we would encourage investigators to mine them and start to put them into their own studies so we can move toward more effective treatments. In order to do that, data access can be a bit tricky, but it is similar to what is needed for other genome projects at the National Cancer Institute, including TCGA, or the Cancer Genome Characterization Initiative, or GWAS studies. So here are some URLs just to provide a base if you need the information. So it's important to note what the data access policy for Target data is. Target aims to accelerate the process of discovering therapeutic targets for which researchers can develop effective new drugs and treatments to ultimately improve outcome for children with cancer. The only way we can do that is by getting multiple investigators to look at the data and work with it and provide greater insight than even we as a fairly large collaborative network can do. The data is made broadly available. However, we do not want to compromise the privacy of the child research patients in any way. So great care is taken in accessing this data and who can access it and for what purposes. Patient privacy is of the highest priority to both NCI and target collaborators, and the genomic data is protected in 2 tiers. The open access tier is available for any data that is not possible to identify any patients, whereas the controlled access tier contains broader demographic, clinical, and genotypic information. But again, that data will not ever directly contain patient identifiers. So the first level, as I mentioned, is the open access tier. It is publicly accessible to anyone. You would be unable to generate a data set unique to any individual from that data. Some examples include HIPAA de-identified clinical data, tissue pathology data, gene expression data, unless it's HUX, as it could be genotypic, so that level 1 data is still protected. Tumor-specific copy number alterations and loss of heterozygosity, DNA sequence data of single amplicons, and tumor-associated mutations. The controlled access tier contains everything you can't get with the open access tier. It is a broader demographic. The clinical and genotypic information that is specific to an individual and possibly identifiable is kept controlled, although all other clinical data is available publicly in the open access tier. And data, regardless of being in the controlled access tier, are stripped of direct patient identifiers. And examples of this would include specific demographic or clinical data, as previously mentioned, genome-wide genotypes for each case, information linking all sequence traces to an individual, whole genome, exome, or transcriptome sequences for any individual case. Now, there are 2 ways to access protected target data, which can get confusing, so I've made sure to include all pertinent URLs and tried to list out information that would be helpful for investigators. First and foremost, to obtain any controlled tier target data, you need to apply for authorized access through dbGaP. And your project needs to meet the following target data use limitation. These data may be used for biomedical research by qualified investigators that are performing research projects that can only be conducted using pediatric data, i.e. the research objectives cannot be accomplished from using adults, and that have likely relevance to developing more effective treatments, diagnostic tests, or prognostic markers for childhood cancers. This data can be used for research relevant to biology, causes, treatment, and late complications of treatment of pediatric cancers. Applications proposing methods, software, or other tool development would not be considered acceptable use of the data. Now, this last piece is under revision at the NCI, and going forward, patients will be consented in a different format so that hopefully we can loosen this data use limitation slightly, but we will never loosen it to a point that does not completely protect the child participants. This is the page that you would go to apply for dbGaP authorized access, and the instructions can be found within. It's about a 2 to 4 week process, and once you gain approval, then instructions are sent. So the data matrix is the primary source of all target data. And I'm sorry I skipped through this quickly, but if you go to the target landing page, the URL has been provided. You can just quickly link straight to the data matrix, which is basically in tabular form and outlines exactly what is available. And if the data is not publicly available at this time, then the links have not yet been put up onto the matrix. So this is versioned so that users can easily follow which time point they obtain their data from for publication. It might be a little bit hard to see depending on your screen, but you can see that there is something called DCC open versus DCC controlled. The DCC stands for the Target Data Coordinating Center. It is where all of the chip-based data is housed and most of the upper level sequencing data. If we're able to make it open access, we do. And you can follow those links to those. But as I mentioned previously, anything that does need to be controlled can be accessed through the DCC controlled link, and then you can drill down to specific levels of data that you need access to. So one important thing to note is the clinical data. You can always find that here under the patient data. There are different types of files that would be available. For instance, if you went to the clinical file, you're going to be taken to an FTP directory, and just as a note, All of the data that is housed at the DCC will be found in an FTP directory that is set up the same as this one is, and it is clearly labeled. You can just go through each piece and find what you need very easily. The second part of this is a case matrix. So here we have provided in a somewhat Excel format so that it's sortable by the user, what data is available by each case. And what will be coming when all of the data is released somewhere in the spring of 2014 is a sample matrix that delves one level deeper. And so even by each sample within that case, so as I mentioned previously, many of our samples will have diagnostic tumor, matched normal, and relapse samples for that one case. And they may have different amounts of data depending on how much DNA or RNA we were able to obtain. So that way users can quickly identify subsets of patients they may want to analyze. So target level, or target data, excuse me, is provided in 4 levels. They are the level 1, which is the raw or trace data. Level 2 is processed or normalized data. Level 3 is aggregated data, which is that is segmented or interpreted, things like gene lists. And level 4 is cumulative data that's a summary analysis result, and this generally comes out following publication. Here is just a little bit more detailed list of the data levels and what might be found. For instance, in the level 1, you can find cell files or IDAT files and even sequence trace files. The level 2 or processed files are the loss of heterozygosity calls, probes, locus amplification or deletion in a sample. BAM files are considered level 2 data. Level 3 data, segmented or interpreted, would include for sequencing, MAF or VCF files. If they are verified, then they are open access. If they are not verified, then they are still controlled access. Also, expression profiling or determination of amplification in a case. And finally, level 4 would be discovery that genomic regions are amplified in 10% of target cases, and things of that nature that are generally published in manuscripts. So as I pointed out before, you can go straight to the target data matrix, and that is the preferred way to get at the data. But the only data that is not physically housed within the NCI's Target Data Coordinating Center are the BAM or trace files for the sequencing, and those are housed with NCBI, either in the trace archives or SRA, the sequence read archive. And it does require the same dbGaP authorized access, so that process can be done at once for both. However, if you'd like to find out what is available through SRA, you can go to dbGaP and search for target. You can either search for the parent project, or as you can see on the right hand side, I have circled, you can search by sub-project. We have all of the target sub-projects listed out so that you can determine what's your point of interest. If you go to the dbGaP page and you actually scroll down the page, there'll be a link to other NCBI resources, and one of those includes SRA directly, or you could go straight to SRA through NCBI. And if you look up target or the PHS accession numbers for target, you will come up with a list of what is available. By the right hand side, you can look for sources of RNA or DNA. You can also look for the type of sequencing, be it genome or exome, and determine what is available. So at the sequence read archive, again, it's our current system for storing BAMs at NCBI. We will also have BAMs available through CG Hub in the future, the same way they are for TCGA. The SRA website itself does provide information on the samples. Anyone can access the information on the samples. However, they cannot actually access the data files without navigating through the dbGaP website, and that's where things do get confusing. You will need an Aspera download client for downloading any actual sequence data files. And controlled access, once you have controlled access, you will be able to sign in the same way you did to request your access, but you will go specifically to the project, and I'll show you more about that in a minute. There is XML metadata available at SRA, which is, there's a parsed version at the DCC. And now that we have sub-studies, if you're interested in just a specific subset of target data, as I said before, you can go straight to that, or you can get all of the target data. Controlled access, importantly, gives you access to everything generated by all 5 target disease project teams. So again, this was just from the SRA website. This is public facing, just to show an example. You can pull up the neuroblastoma whole exome sequencing specifically. It does tell you that that was done at the Broad Institute. If you click on that link, you can go specifically into a single run and see all of the information surrounding that, what sample was used, what submission it was in, anything you would need to know metadata-wise. And just to get through the identifiers, all of these runs and experiments and other metadata are stored in SRA numbers that could be difficult to interpret. So here I've just provided you with a list of what these accession numbers stand for. So if that gets confusing, you know what that means. However, as I mentioned, you cannot get BAMs without a special download client. You do need the approval through the dbGaP to get to the target data. This does need to be renewed every year. You can download and install a spare client locally on your own computers, and if you navigate the dbGaP website and download appropriate files, you currently, to a degree, need to look up IDs, but this is being changed right now at NCBI to try to make the process a bit easier. And our target DCC personnel are happy to provide detailed instructions for how to access this. But quickly to go over it, I showed you the login page earlier, but if you want to go through the dbGaP interface so that you can learn more about how the database for genotypes and phenotypes works, if you come to their main page, you will be able to go to apply for control data access, and it will take you to where you needed to go previously to log in and to navigate here to My Requests. So My Requests is how you put in the request for approval. But when you're actually going to download the data, what you will go to is My Projects, because approved users already have this loaded up into that tab. It will look similar to this, and you'll be able to just click on the target data. And what you'll find is something similar to this. This is NCBI's new run selector, where you can actually sort by the information that you'd like to obtain. So if you specifically want, this is an example again from Neuroblastoma, which is PHS number 467, but you'll note that each of our sub-projects has a different 3-digit number at the end, and the target's parent study is 218. So if you want to sort by specifically whole exome sequencing that was done at the Broad, you can do that. If you want to sort by males, you can do that. Sort by tumor samples or normal samples, and that way you can get the data set that you're looking for prior to actually downloading. I can tell you, if you do not do that in advance, downloading this material, if you try to get everything that's within target, it's close to 3,000 data files, and it will take a lot of storage and about 3 months to download. And this, you can see a bit of a tree view that you can also use to get at the data you want. If you do want everything, you certainly can. And then you would just, rather than using the run selector to select a subset of cases, you can just go through the tree view and check everything. Again, you must have Aspera downloaded, and you can check on the data manifest if you would like for what is available. Again, this is going to be thousands of rows, so it may not be the most efficient, but if you have a script for a different way that you'd like to parse, you could use it on this data manifest. So in addition to Target, there are 2 other current programs ongoing in the Office of Cancer Genomics, including, as I mentioned before, another large-scale initiative similar to TCGA but studying different cancers as the Cancer Genome Characterization Initiative, as well as a more translational program that pulls together the data generated from these large-scale projects, and that is known as the Cancer Target Discovery and Development, or CTD squared network. And just very briefly, CGCI is a multi-project initiative, and there are some completed projects that we're looking at Myeloblastoma and non-Hodgkin lymphoma. The publications from those, I have listed initial publications here. There are other publications that can be found at the website listed on this page for more information, but all of that data is also available. You apply for it in the same way that you apply for Target data, or for that matter for TCGA data. Even though it goes through CG Hub, you still do use the dbGaP process. For ongoing programs in CGCI right now are the HIV positive tumor molecular characterization program, which is studying lung cancer, diffuse large B-cell lymphoma, and HPV related cancers in HIV positive patients. As well as the Burkitt lymphoma genome sequencing project, which is looking at Burkitt lymphomas, many of them from overseas in Africa. The cases are being characterized as follows, with mRNA-seq of the tumor RNA, with DNA sequencing, whole genome or exome of matched tumor normal DNA. And again, verification of candidate mutations is going to occur, same as it will with Target. And as I mentioned, CTD squared is meant to bring all of this large-scale genomic data together and rapidly translate it for the patient benefit or precision medicine. This is surrounding high throughput identification and characterization of potential targets for cancer therapy, be it genes or pathways, as well as identification of small molecules that modulate validated cancer targets found through either the CTD squared network themselves or through these large-scale genomics initiatives. There's an emphasis on interactions of centers, so the CTD squared network was piloted as a 5-center network that did publish their results, and the data was so successful with the team science and with the approach that the network has been expanded to 13 centers, and those centers will focus on bioinformatics, genome-wide loss of function screening, targeted gain of function gene validations, mouse-based in vivo screening, protein-protein interactions, and small molecule high throughput screening. The CTD squared network will share validated candidates with the cancer community, allowing investigators to contribute to the understanding of the mechanisms of cancer and potentially accelerate development of clinically useful markers and therapeutics for evidence-based treatments, as is the goal with all of our projects discussed here today. And as I mentioned, the ARA funding allowed for the expansion of the CTD squared project into what you have here, and each center has different disease expertise. You can go to the URL listed on the previous page to find out more information and use this data. So with that, I would like to acknowledge our NCI staff in the OCG program office. Dr. Daniela Gerhardt is our director. And I, of course, manage most of the pediatric programs. Dr. Megan Stein is another program manager in our office who is managing the CGCI efforts. The Cancer Therapy Evaluation Program, Dr. Malcolm Smith, is co-directing the Target Initiative and certainly was key in its inception. Our OCG communications team, Drs. Shannon Berman and Jessica Mazarek, and of course our OCG Data Coordinating Center team who handles all of our data, Dr. Tanya Davidson, and senior bioinformatics engineers Patty Yesuan, and Leandra Hermida. I thank you all for your participation, and I hope that you will visit our site and use our data and help us to come up with ways to treat our patients. And if you would like to obtain continuing education credits, please follow the link on this page. Thank you.